Hello and welcome back to the Biochemistry for Health Sciences channel, the BHS channel. In today's video, we will continue with lipids and we'll talk about a little bit about an overview of another class of lipids that we call steroids. So there are many different steroids present in our body. The best way to recognize a steroid is to look at these four fused rings. Okay, so we have a six-membered ring, a six-membered ring, and a five-membered ring, all fused together. So this four fused ring skeleton gives steroid its characteristic structure. Probably the most important steroid we all heard of is cholesterol. And from cholesterol, we make all the other steroids, and that includes the steroid hormones such as cortisol, aldosterone, progesterone, as well as the estradiol and testosterone. So these two come from the adrenals, while the rest are the sex steroids. Bile salts are also steroids. So a little bit about cholesterol. We all probably heard how important cholesterol test is in our serum. So that's one of the tests that forms part of what we call the lipid panel. So typically cholesterol, triglycerides, and then the various kinds of cholesterols, HDL cholesterol, LDL, VLDL, as well as all kinds of ratios are sometimes measured or calculated in the lipid panel test for serum lipids. So if you look at the structure of these different steroids, you can see basically the differences between one steroid and another steroid is the differences either in the ring, for example, where is the double bond placed in the ring, or differences in the substituents on the rings. Okay, you can see that substituents on the rings differ from one steroid to another different steroid. As I mentioned earlier, bile salts are also steroids. Okay, we'll talk about the function of bile salts in a few seconds, but this is what is stored in bile, which is in the gallbladder, produced by liver cells and then stored in gallbladder. So serum cholesterol is a common test, so good to know a little bit about the nature of cholesterol in serum. Cholesterol is not water soluble, so it is transported in serum in the form of lipoprotein. So it's transported in the form of lipoproteins. So there are two types of cholesterol in these lipoproteins. One is called free or unesterified cholesterol, that's about 30%, and the other is called cholesterol esters. So the difference is you can see the hydroxyl group here has a fatty acid, any fatty acid that is attached in a cholesterol ester. Free cholesterol doesn't have fatty acid attached. So clearly this is much less water soluble because of its fatty acid. This is slightly more water soluble because of a hydroxyl group. So in a lipoprotein, you can see the cholesterol that is unesterified, free cholesterol, tends to be found more on the surface because of that hydroxyl group. But in case of the cholesterol ester, it's found more deeper inside the lipoprotein sphere, spherical particle. So free cholesterol tends to be found more on the surface. Cholesterol ester tends to be found more inside the lipoprotein particle. So cholesterol is a component of all the different types of lipoproteins. We'll talk about lipoproteins in a future video, but there are different types of lipoproteins. Chylomicrons has cholesterol. Very low density lipoproteins or VLDL has cholesterol. Low density lipoprotein or LDL has a huge 50% cholesterol, and the particle HDL or high density lipoprotein has about 20% cholesterol. So cholesterol is found in all these different types of lipoproteins. The function of cholesterol, as we mentioned earlier, is part of membranes, and cholesterol plays a very important role in maintaining the fluidity of membranes. Okay, it can increase or decrease the fluidity of membranes. So for example, when the temperature is higher temperature, that tends to make these phospholipids move around more. Okay, because the higher temperature, the increased energy will make these phospholipids move much more and make it behave more like a liquid. Okay, so higher temperatures tend to make the membrane more like a liquid, just like butter melts, becomes more like a liquid at higher temperatures. In that case, the cholesterol tries to prevent too much of this membrane becoming liquid. Okay, so basically, because it is strongly interacting, you can see the OH here is interacting with the polar group, while this rigid ring, this rigid ring is interacting with the non-polar part, and then the more flexible non-polar part is also interacting with the membrane lipids on either side. So at higher temperatures, cholesterol tends to prevent the membrane from becoming too liquid. Now at lower temperatures, when the temperature decreases, that low temperatures tend to make lipids more solid. So melted butter is an analogy. A melted butter will try to solidify when you decrease the temperature. Once again, now here in this case, the solid means less space. So this membrane lipids will tend to come closer to one another at lower temperatures. But once again, that is prevented because you can see the ring, the rigid ring as well as the flexible non-polar chain of the cholesterol is preventing the membrane lipids from coming together. So at low temperatures, cholesterol makes sure that the membrane doesn't become too solid. So this effect of cholesterol in making sure that the membrane is not too solid or the membrane is not too liquid, that's an important function of cholesterol in membrane. Now you might say, why do we worry about temperature? Because our body is always at 37 Celsius. Think about it, that's actually only true at the core body temperature, but at your extremes, your fingers and your toes and so on, the body temperature can be much lower or much higher. So steroids like cholesterol are very critical for membranes, but steroids such as the steroid hormones, these are signals, very important signals in cell signaling. So basically the signal steroid is not water soluble, so it basically is transported into the membrane rather than binding to a receptor on the membrane. And we'll talk about it under cell signaling. So the receptor is inside the membrane. Steroid binds to the receptor and then the whole thing goes into the nucleus. You can see an example of testosterone here. The whole thing goes into the nucleus, and the steroid receptor complex attaches to the DNA and affects how the DNA, how fast the gene is copied into RNA and eventually into proteins. So basically that cell signaling will affect the cell's protein profile. It will change the protein profile of the cell. And since proteins do most things for the cell, that will change the cell's activity. Okay, so steroid hormones are very powerful signals because they go right to many genes. Okay, they can affect many genes and change the protein profile of the cell. And again, we'll talk about this under cell signaling. So bile salts are also made from cholesterol. So bile salts such as glycocolate, taurocolate, cholic acid, and so on. These are all also made from cholesterol in your liver. And then they are sent into the bile duct to your where they are stored in the gallbladder. And once in the gallbladder, you have your meal. The gallbladder squeezes its juice out, and the bile salts get out into your digestive system. So in the small intestine, bile plays a very important role, bile salts. What it does is when you have fat particles, you have eaten a meal containing fat, those fat particles are basically emulsified or broken into smaller particles. Smaller fat particles have larger surface area. And then the pancreatic enzyme lipase, which sits on the fat particle. You can see the green lipase sitting on the fat particle. It's a surface active enzyme. It sits on the fat particle. So because there's a higher surface area, more lipases can come and sit on this fat particle and break down the fat particle into glycerol and fatty acids. So bile salts increases the surface area of fat particles by emulsifying the big fat particle into smaller fat particles. And that allows lipase to digest the fat into glycerol and fatty acids. So clearly, if there is insufficient bile salts, anything that is not only is going to impair the absorption of fat, but also in the fat, there is fat-soluble vitamins too. And that absorption of fat-soluble vitamins will also be affected. So bile is stored in the gallbladder. So in the bile, you have not only bile salts, you have a lot of cholesterol, and of course, then you have some amphiphilic lipids. So if there is too much cholesterol in the bile and not enough bile salts, then the cholesterol can precipitate because it forms a supersaturated solution. So the cholesterol can precipitate out of the bile, and that can lead to the formation of gallstones. Okay, and again, this will be a topic that we will discuss in the future, since it does involve quite a bit of chemistry and biochemistry, the formation of gallstones. So bile salts are very important also to make sure that we don't get gallstones formed in our gallbladder. Okay, so that is it for now. A little bit about just an overview on steroids. Hopefully we will see you in the next in the next video. So please do not forget to subscribe to this channel so that you would be able to see the previous videos, as well as many more fun and exciting videos to come in the near future. Again, these videos are meant to connect chemistry and biochemistry, as well as cell and molecular biology, to medicine. Okay, so basically the purpose of these videos is to help healthcare professionals connect molecules to medicine. Until next time, goodbye.